today, I also wanted to give you some information on this particular microbe, which is the causative agent of Lyme disease. It is called Lyme disease after Lyme, the city in Connecticut, where it was first identified in the 1970s. So the causative microbe is Borrelia burgdorferi, and it is a bacterium. And as you probably know, this is a bacterium that can infect ticks, as well as many different types of mammals and also some amphibians. And this disease causes an initial infection that is quite noticeable in many individuals, but in other individuals it's not noticeable at all, so it's asymptomatic in some individuals. And then regardless of whether or not the initial infection was asymptomatic, it can be followed by a period of latency and then reactivation. So early on, the first indication of the disease usually can be a rash, and so this is a characteristic bullseye rash that was once thought to be the defining characteristic or the best sign to tell whether or not an individual had been infected with Lyme disease. Now, however, we know that more than half of the cases of individuals do not notice a bullseye type rash when they are infected with this pathogen. So this is no longer used as the most reliable indicator. These ticks, as we've said, are found in the United States, and many of the areas where they're found are nearby. So they include the Northeast area, as well as the mid-Atlantic states, and all the way north up into parts of Maine. And then there are also the ticks that carry this disease found in the central northern part of the U.S. Now, there are also similar bacteria. They are also Borrelia, but they are different species. So you can see down here. And there's two types, and these types of bacteria are carried by ticks in Europe. Now, in the US, there are predicted 40,000 cases per year, and we think that that is underreported or underreporting the number of cases in the US total because people, again, are often asymptomatic initially. Now, this infection, although it might not be severe early on, can become a huge issue because as the pathogen reactivates, it can start to have an effect like arthritis, so it will cause pain and stiffness in the joints. It can affect the connective tissue, and then eventually it can also begin to affect a person's memory, as well as their muscle control. So these are long-term effects if the disease is not treated. But as I said, sometimes people don't know they've been infected, so it can be difficult, and people in the recent past definitely have gotten to these stages where it's really kind of mysterious what is causing these strange and severe effects, such as muscle loss and confusion and loss of muscle control or movement, but it is due to the bacterium. So in addition, this bacterium, although it's gram-negative, is relatively difficult to treat, so you need long courses of antibiotics, multiple antibiotics, to get rid of this pathogen because it's really good at evading our immune system, and the antibiotics just don't work as well on it. It's not highly susceptible. So as we've been saying, and as I'm sure everyone knows, this disease is carried by ticks, and in addition, so down here we can see the different hosts. So this is the black-legged tick here, and this is what carries the Borrelia, and this is the Borrelia here. As you can see, it's a really interesting cell shape. It has a spirochete shape, which means that it's wavy, wiggly, and this pathogen can infect mammals, including humans, deer, fox, cats, dogs, possums, squirrels, chipmunks, mice. It could also infect birds. And we'll talk about why this is relevant in a couple minutes. I also wanted to point out that the ticks that carry and spread this pathogen have different stages of their life cycle. So you may have seen this comparison in the past. On the left, we see an adult tick. So this is probably more than twice the size of a nymph tick, which is a much younger tick. And this is on the right is an engorged tick, which means that this would be feasibly the same size starting as the one on the left, but it has taken a blood meal, basically has eaten as much as it can from someone or some animal. So the problem with this is that the ticks can be very difficult to find and see on your skin, so they're far smaller than the size of a penny. But the good news is that the larger the tick, the easier to see, and the smaller the tick, the less likely they are to be infected with Lyme disease. So that means that it's easier to see the ticks that might carry Lyme disease. And the reason for this is because the ticks have to pick up the Lyme disease from a host. So when ticks first hatch from eggs, they will not have the disease. They can't start out having the disease. They need to take their first blood meal as a larva. So basically it looks like a very, very small version of the adult. And if they attach to a host that has the disease, then they will get the disease, they will start carrying this bacterium, and then they could transmit it to any subsequent hosts that they attach to. So really, later, the older the tick, the larger the tick, and the older the tick, the more likely it is to have the Borrelia pathogen, because it's had more blood meals, so it's had more opportunities to attach to a host that has this pathogen. And also, the later in the season, so in the fall, you tend to get more ticks in the population that actually have this disease. And I say that they have the disease, really they carry the disease. We don't think that the bacterium negatively affects the tick. It just kind of hitches a ride, and it is then transmitted to other organisms that the tick latches onto. And I was talking about all the different types of organisms that can be infected before, and this is relevant because the ecology of an area impacts and correlates with the transmission of the disease. And so there is a lab at the Carnegie Institute in New York, and they've been studying this for years. And they've looked at the size of fragmented forest pieces and the diversity of the organisms in those fragmented forests and the prevalence of disease in those fragmented forests. So in other words, they've looked at places like intact forests. They've looked at places that might be cleared lots that have, grass, they're empty lots, but they're adjacent to forest. They've looked at areas that are more developed and They may or may not be adjacent to forest, as we see here, so this is not adjacent. And they've looked at things like how many white-footed mice are in that area. So if you think about it, the less woodsy an area, the more likely you are to have mice, probably as the most common type of mammal. Probably also squirrels, but mice are there. If you've ever had issues with mice, you know that you might not be seeing them, but they are there. And mice are really good at living with humans in their homes, and they don't need a lot of intact forest. So white-footed mice are really good, again, at living in these fragmented forests. And they're probably the most common, or one of the most common mammals in highly fragmented forest and, areas that are urban. What does this have to do with the ticks? Well, the white-footed mice are often infected with ticks, and unfortunately, the Borrelia pathogen replicates really well in white-footed mice. Now, we haven't talked in the past about the success of pathogens in different hosts. This pathogen, the Borrelia, does not replicate to the same population levels at the same rate in every host that it infects. The white-footed mouse is one that the bacterium just replicates really well, really quickly. You get really high concentrations of the bacteria in a mouse. And the mouse could still survive. On the other hand, there are other organisms, even deer. We might see deer that have a lot of ticks on them, but the Lyme disease agent is not going to get to really high densities in the deer's blood, just because they're not a great host for it. So let's take a look at this graph, or these graphs here. So these are from that lab I was talking about. The principal investigator, or the boss of this lab, is Richard Ostfeld, again, and this is in New York, and they looked at the density of nymphs, so young ticks, and specifically the type of tick that carries Lyme disease, which is the black-legged tick, compared to the size of the forest fragment. So the label got cut off at the x-axis here, but this is the number of hectares that the fragment was in total. And so we see the smaller fragments of forest have many more nymph ticks in them, which is interesting. The density is much higher. And similarly, the density of the tick larvae, a different life stage, is higher overall in smaller areas, or smaller fragments of forest. Now we also see that there is a correlation between the number of tick larvae that are found on each white-footed mouse and how many chipmunks there are. So it's not just the size of the piece of forest, but also what other organisms are there. So if there are a lot of chipmunks, then the ticks kind of tend to disperse. So we could see that if you just have mice and very few chipmunks, then the mice end up getting all of the tick larvae. But as we increase the density of chipmunks in this area, then the number of tick larvae per mouse tends to go down. So the chipmunks kind of help to carry the tick burden. And fortunately, having chipmunks there actually will decrease the likelihood that organisms are going to get Lyme disease through this tick. Now let's take a look at this graph. So this is the proportion of larval ticks that were fed, And so this was a great study, and I actually heard one of the investigators present it at Wesleyan, and this data was gathered by actually capturing these animals, so taking mice from the wild, from some certain area, and keeping them in the lab. They fed them, they were comfortable, but they kept them in the lab for a week or more until all of the ticks that could possibly be on them had dropped off. So the ticks had fed, and then they subsequently were dropped off of that organism, and then the ticks were collected and counted. And in this graph in particular, they're only showing the larval ticks. So mice had the highest proportion of larval ticks in total. And then birds, the very is a bird, and then the catbird, they had a much lower proportion. Chipmunks, again, had a much lower proportion. And squirrels, so these guys, you see that they all have a B at the top. That means that they're statistically similar to one another, but they're different from the mouse. So the mouse has significantly more ticks than any of these groups in the middle. And then the champ here is the possum. The possum had the lowest proportion of larval ticks fed. And that is actually because possums will eat the ticks off of themselves. So if they get a tick, they'll pluck it off, usually before it could feed, and they will just eat it. So possums are really awesome. I don't think we appreciate them enough. But also, we tend to see more possums in larger fragments of forest and where there's more diversity overall in terms of animals. And so again, that you might see possums, in a residential neighborhood, but you don't see one on Main Street in Middletown, for example, or a place that's even more urban than that. And so this lab came up with this schema, and they have done many, many publications to support this. So this chart shows that, this diagram shows rather, that white-footed mice are not as good at grooming off ticks compared to possums. So possums are really good at grooming off ticks. You can see that the white-footed mouse here will groom off and kill a number of ticks. So they do groom themselves. If you think about a mouse, they do clean themselves, but they're not really good at getting those ticks off. A lot of the ticks, pretty much the same number of ticks that they take off, are managed to stay on and get a blood meal, and potentially will transmit the disease if they have the disease, or pick up the disease if they don't have the disease. So we see about 1,000 ticks were taken off and killed, and then 900 ticks were fed and infected, and then 78 were fed and uninfected. But then we compare that to the possum. Look at this possum and the job that it does. It groomed off and killed 5,487 ticks. That's a lot. And then ticks that were not taken off and killed or eaten, that proportion, many, many more were given a blood meal and fell off and were not infected from the possum. So that's 194 versus the number of ticks that had a blood meal from the possum and ended up being infected with Lyme disease. So this is just showing these two mammals, but what this lab has found is that the most crucial animals to look at in this system are the white-footed mouse, because they're unfortunately so good at providing a home, basically a host for the Borrelia pathogen, and possums because they're so bad at replicating and providing a host for Borrelia, and because they destroy those ticks. So the takeaway here, I guess, is that ecology can be closely correlated with the prevalence of disease. So we need to think about ecology when we talk about disease. And also, we should say thank you to our possums.